And so if it's Wednesday, it's time for a Learning Farms webinar. And so we are really excited to have Dana Kolpin. Dana is a research hydrologist with the Central Midwest Water Science Center in Iowa City, Iowa. Dana has been with USGS since 1984, and his research interests include the fate, transport, and effects of environmental contaminants in the environment. So we're really fortunate to have an expert such as Dana here in Iowa, and certainly excited for his important talk. He's going to talk about a comprehensive statewide spatiotemporal assessment of PFAS in an agricultural region of the U.S. As always, if you have questions as we go along, please type those in the chat box, and I will get those to Dana at the end of his talk. With that, I will turn it over to Dana.
All right, thanks, Matt. So let's get started here. Actually, we'll advance the slides, repeat the title slide just to show my collaborators on the study, both from the USGS and from the University of Iowa's Center for Health Effects of Environmental Contamination. So before I get started, I thought just making sure everybody's kind of up to speed on what PFAS is. What's this talk all about? PFAS are a class of man-made chemicals, a very large class made up of more than 4,700 chemicals. And really, they're much more, way broader than the PFOS and the PFOA that most of you have probably heard about. All these PFAS have extremely strong carbon-fluorine bonds. These are one of the strongest bonds in nature. They have a whole range of beneficial properties that lends themselves to a whole range of products. They have oil and water repellency, temperature and acid resistance, friction reduction. Again, these properties lead them to a whole range of products. That's from paints to food, fast food packaging. They're in the inside of your pizza boxes, so the pizza doesn't stick to the lid. They're inside of microwave popcorn. They can be in dental floss, whole range of things that many of us don't even aware that they're in there. But it's these beneficial properties that these PFAS have also lend them to cause a variety of environmental concerns. These extremely strong bonds make them very persistent, and that's the reason why they're called and coined forever compounds. And we're now realize that they're pretty much found in every environmental compartment we've looked for. They're extremely mobile, so they're found in areas far removed from their source and manufacturing. They bioaccumulate. That's a really important aspect in terms of exposures and environmental consequences. And really, they're found in a variety of organisms, including humans. If all of us donated blood today, we almost all of us would probably have some levels of PFAS in our bodies. And there's known and suspected toxicity, not only to environmental organisms, both aquatic and terrestrial, but even in humans. Just cancer and immune dysfunction are just a couple examples of some human issues. Now, today, most of the PFAS research has been related to urban pathways, and that's related to wastewater treatment plants and their discharge, whether that's effluent into streams or sludge or biosolids that are land applied. In terms of contamination around airports and military bases due to firefighting foams. But there is agricultural sources as well. These are really kind of less known and less understood. And these ag sources can come from land application of biosolids. Again, that biosolids is kind of that sludge that's come out of wastewater treatment plants. There roughly half the biosolids generated in the U.S. are land applied for a beneficial source of nutrients, but they also can contain a host of contaminants, including PFAS. There's been PFAS to be shown to be present in livestock waste. This particular study that I cite here found that this is a study from France that livestock waste did have PFAS in the manure that they looked at. Now, this is the same study that also looked at biosolids. So yes, livestock was a lot less than the concentrations they found in the biosolids, but it wasn't zero. So that still could be of consequence once we start putting livestock waste all across the landscape. And even pesticides can be a source of PFAS. I caught a talk recently that stated that I think since 2015, 70% of the new pesticides being registered are in fact PFAS type compounds. So it's not only active ingredients, but a number of the inerts that's in the pesticide formulations can also be PFAS. And I've even seen papers that show that PFAS can be in pesticide containers. So, the inside of the containers are coated with PFAS, so the products slips out, and so all the product comes out of the container after it's used. But that use can also then bleed or leach the PFAS into the formulations and then gets out into the environment. So the whole range of potential agricultural sources as well that really haven't been thought about near as much as the urban aspect. So that kind of got me to lead to thinking, it's like, okay, so what are the levels of PFAS in these rural agricultural streams? We see these streams as we drive across Iowa all over the place. We have farming pretty much up to the banks of these streams, but we really don't understand, are there PFAS in these streams or not? And given Iowa's intense agricultural production, it's really an ideal landscape for determining the prevalence of PFAS in these rural type agricultural streams. Now, before we jumped out into investigating these streams, it's always good to take a look and see what historic retrospective data may be available. And certainly the U.S. Environmental Protection Agency through their UCMR 3 did look at PFAS all across the country. And there was, in fact, 82 drinking water sources sampled in Iowa. And when you look at the map of those results, you'd say, nothing to see here. They really didn't see any PFAS in these settings. But if you look at the data a little bit closer, almost all of the drinking water sources were in fact from aquifers. So if you're trying to get a better understanding of PFAS in streams, well, data from aquifers is really not going to tell you that question. And the streams, the rivers that were sampled were actually the larger rivers, because the EPA was looking at drinking water sources. Obviously, the drinking water intakes in Iowa are only on the large rivers and streams. So still, those small rivers and small streams that I provided an example on the previous slide really seem to be falling through the cracks. And the other limitation for this particular retrospective study is, at that point in time, they only looked at 6 PFAS. And we know, like I said, there's a huge class of compounds, so 6 is a pretty small number. If you look at the reported limits, at the time it was fairly decent, 10 to 90 nanograms per liter. But again, that's not as sensitive as they would like to see, because these compounds do bioaccumulate. We like to get these levels down as low as we can get them. So with looking at this database, I still felt like more PFAS data is needed to really better understand and determine the prevalence in rural agricultural streams. But before we could go out there and jump out and do such a recon, we needed a method. We needed a method to be able to measure these compounds in water. Unfortunately, the USGS had recently developed such an analytical method, looking at 34 PFAS, still, barely scratching the surface, just the tip of the iceberg, but still about, 4-fold more than that previous US EPA study looked at. And that included both precursors and PFOS and PFOA replacements. And that includes compounds like Gen X and Adona and so forth. Reported limits were lower, generally lower than what the previous study shown. Still room to be, there's room for improvement. I'd like to see our reported limits get down to at least a nanogram per liter, if not lower. But we're getting there. We're improving. We're already improving this method with time, and we'll get that sensitivity down lower than we had here. And the other important thing, and I'll bring this up later as well, is this particular study, we included semi-quantitative results as detections. So they met all the criteria for a detection. The actual concentrations are a little bit fuzzier because they're down there, kind of down close to the weeds, but they are real results, real detections, and we included them as such. Now, the one good thing about this method, I won't go into the details other than to say, these samples, it really just takes these tiny little 2 milliliter centrifuge tubes that you fill half full. So they're very easy to collect in the field. There's no filtration. You just, whether it's weightable, you just go in the middle of the stream and dip and fill the tube half full, snap it shut. They're small. You can put tons of these things in a single cooler versus a 1 liter sample like we often have to do for other water quality results. That takes, you can only fit 7 or 8 in even a big cooler. So this is much easier logistically in the field. It's much easier to ship these things. But the one thing about PFAS, because it is everywhere, it's in the equipment we use, it's in the equipment, a lot of the equipment the laboratory uses. It's also very easy to contaminate these samples. So it requires a lot of field QA, QC, particularly the field blanks, to make sure that there's no field contamination or contamination of the sample after the collection. Okay, so once we had the method, now we're off and running to do a spatiotemporal sampling in Iowa stream. So what we did here is we did an opportunistic, we call an opportunistic sampling over about a 7-month period, which means basically because the USGS is going all over the state for a variety of reasons to visit our USGS gauging stations for routine maintenance and discharge measurements, to collecting water quality samples. So basically we asked our field personnel, when you're out in the field to these sites, can you collect us these small vials, put it in a cooler, ship it back to us. We'll throw it in the freezer and see what we get after into this period. And that's how we really did what I feel is kind of a low cost mechanism for getting a really nice statewide data set. And what we did then is once we had the samples in the bag, went to our colleagues at Cheech, University of Iowa, and say, these samples are collected. All we need now is help with the funding for these analysis. So they were more than willing to help with the funding of the analytical costs. So really, we effectively got a fairly expensive, if you had to start from scratch, statewide assessment for really extremely low cost. Ultimately, we had 119 samples collected across 60 streams. These streams cover 88% of the basin across Iowa, and they cover a range of basin sizes, cover a range of land use, both agricultural and urban land use, and they cover a range of densities of both livestock and people. So really feel like we kind of captured a representative sample to get an idea of how prevalent these compounds were across the state. So when you look at the results, and these results have been published both in journal article and ES&T letters, and what we call our USGS data release. That's where the raw results are. In fact, we found PFAS in 26 of the 119 samples collected. So that's really kind of good news and bad news. The good news is, we didn't find these PFAS in every stream that we collected, in every sample that we collected, but we did find them. So now let's dig into this a little bit deeper. Of the 34 PFAS that we measured, we had 10 with at least 1 detection, and these are the ones that we were most frequently detected with this sampling network. The highest individual PFAS, 134 nanograms per liter of PFBA, was in fact in a small stream in western Iowa. It's an agricultural stream, so that shows that, yep, these small streams, doesn't mean that every stream is going to have PFAS in it, but we did find, in fact, that there was PFAS in some of these small rural streams that we were interested in. And that semi-quantitative nature that I mentioned previously really is coming into play. 42% of the hits that we measured were of that semi-quantitative nature. It's a really low level. And we did get some pushback to say, why are we presenting that? Well, we should be just calling those non-detections that were kind of sensationalizing results. But in fact, we feel these semi-quantitative hits are very important. If you exclude those, you're underestimating exposures. And because of that bioaccumulation nature, these compounds have the affinity to bioaccumulate. It doesn't matter how low those concentrations are. If they bioaccumulate, they may not mean something to bugs or insects or terrestrial life today, but they could mean something tomorrow or some point in the future as they continue, as exposure continues and those concentrations can build in the various organisms. So again, I think that's extremely important aspect to include the semi-quantitative results in this study. Looking at the spatial distributions, PFAS were detected in 19 of the 60 streams that were sampled. Again, only 9 would have been had detections if we excluded those semi-quantitative hits. So again, I won't rehash that again, but those are very important to include. I didn't see any really obvious pattern to the spatial distribution. These basins in yellow are those basins where at least one PFAS was detected in at least one of the samples that were collected. PFAS was detected in a range of basin sizes from very small streams to rivers as big as the Mississippi River. But the spatial distribution we did see is that the highest cumulative PFAS concentration and the greatest number of PFAS were in fact in what I call wastewater impacted streams. And we'll use Muddy Creek as a nice example. Muddy Creek is a stream about 10 minutes from Iowa City. This is where the city of North Liberty's wastewater discharges into Muddy Creek. And basically PFAS are not currently regulated. So this wastewater treatment plant is not out of compliance. They're just not part, PFAS are just not part of the regulatory equation at the moment. This study, or this stream, we did a longitudinal sampling over a 5.1 kilometer reach. So we collected a sample 100 meters above this outfall. This is North Liberty's outfall. And wasn't completely squeaky clean, but just had one low level detection of PFAS. And you bracket that effluent and go 100 meters of that outfall, go 100 meters below the outfall, and you can see a whole range of PFAS detected. And it's really this contribution, it's this input of the effluent is causing this suite of chemicals. So it really shows, the strong PFAS gradient from the input of this wastewater treatment plant. Now, in this first 100 meters from the outfall to below, you see there is in fact a substantial decrease in concentrations. And most of this decrease is simple dilution from mixing of the native upstream water, which had very, very low concentrations of PFAS. But not all of this decrease can be explained by simple dilution. There's other processes, we can't quantify these processes, but there's other processes taking place, whether that's absorption to sediment as that effluent mixes with the stream water, volatilization, or even foam formation. This effluent, as it discharges into this limestone riprap, as it then cascades about 3 or 4, about maybe 5 feet to the stream, causes this foam to form, and that could have some impacts in terms of decreasing concentrations in the effluent while it mixes with the stream. And as you move 5 kilometers farther downstream, you can basically see the same profile. So again, it's that forever compounds. These compounds can be transported long distances. We do see some decrease. This reach does not, at least at low flow, doesn't have any additional tributaries coming in. So we don't feel that this minor decrease is due to dilution. It's probably these other processes that are taking place. We just can't quantify which ones and which are the most important ones, but you can see that even 5 kilometers away, you're still seeing substantial levels of PFAS across that whole reach of that stream. So looking at things temporally, to look at a temporal distribution, you need some various characteristics. Obviously, you need more than one sample. A lot of the network didn't have more than one sample, but about 50–60% of them did have more than one PFAS sample collected. And to look at trends, you need at least one of those PFAS samples having a detection, so you can see, are things increasing or decreasing? And to really understand how concentrations may change with flow, you would need available flow information. You need a USGS gauging station. So there was 10 sites with these types of characteristics to look at temporal distributions. And of those 10, 7 actually seem to have an inverse relation to stream flow. And again, going back to Muddy Creek, and we sampled Muddy Creek multiple times. This is just one example. I think this was from July. You can see that there is a strong inverse relation. As stream flow decreased, the cumulative PFAS concentrations increased. Now, keep in mind, yes, this is only a very narrow range in flow from just below 2 to about 5 CFS. So will this same trend hold as you get higher flows? That's yet to be seen, but at least it demonstrates that we do have this inverse relation to flow. Now, there's multiple reasons for why there's this inverse relation, but certainly a point source as is happening here in Muddy Creek, that discharge pipe coming out of the wastewater treatment plant is one possible explanation for this inverse relation, where if it was a non-point source, you generally would have a positive relation to stream flow. You need rainfall to kind of pick up contaminants off the land surface and carry it into the stream. But unfortunately, with this kind of convenient sampling, really wasn't set up to understand non-point source contribution. So I'll talk about this later, but really feel we need more targeted sampling to better determine potential non-point source contributions to streams. But again, I'll bring this up a little bit later as well. Looking at ToxCast, of the 10 compounds, 10 PFAS that we measured, 6 had available effects data in ToxCast. If you look at the ERA ratios, this is the exposure activity ratio. So really, this is basically dividing environmental concentrations by the effects concentrations, and concentrations ratios that are above 1 are, known observed adverse biological activity. We didn't have any that exceeded that level, but others have used 0.001 as a threshold of potential environmental concern. Doesn't say there's necessarily definitive effects, but the ratios are such like, that's worth investigating further. And we had a number of samples that exceeded that potential environmental concern. Okay, but one thing I do want to point out with this study, even though it's, it was groundbreaking, it's the first time we have this baseline information available, really because it was only looking at water, this assessment effectively, we feel, underestimates PFAS exposures in streams. Other research has shown when you look at both water and bed sediment, the shorter chain PFAS are more prevalent in the water compartment, and the longer chain PFAS are more prevalent in the sediment. And it's these exposures, the aquatic organisms are being exposed via both water and sediment, and that's how these concentrations and these chemicals can move then through these riparian food webs. So again, we only could do what we could do. We didn't have a sediment method at the time, but this was a good starting point to let's see what's in water and then move from there to get a better understanding. So in conclusion, this study did provide some baseline information that wasn't previously available. We did see that generally the highest individual concentrations are in cumulative concentrations, and the most detected PFAS were in effluent impacted streams. But we did see PFAS even in rural basins. And in these rural basins, there was no obvious urban or industrial sources. So that really kind of begs us to look further to see, what is the source of these PFAS that we're finding. As I mentioned multiple times, these low-level semi-quantitative PFAS detections are very important because the bioaccumulation aspect of PFAS, excluding these semi-quantitative detections, would have effectively underestimated exposures that are important because even low-level exposures could eventually bioaccumulate to concentrations that are have environmental consequences. So what are the next steps? As I mentioned previously, you really need to better understand non-point agricultural contributions. And what that's going to do is you need to move away from kind of convenient sampling, which is, let's just go out when we get there, or to add a kind of a set, let's go out on this Tuesday of this month and see what's out there. You need a targeted sampling to capture periods of land application. So we need to go out when there's either biosolids being applied to the land surface or manure being applied or pesticides being applied. And during that window of applications, then follow up the sampling during corresponding precipitation events. So it's going to take, you got to kind of be ready at a moment's notice because, it could be a night, could be a weekend. You got to be ready for those runoff events to take place and sampling over that runoff hydrograph to see what we see during those types of conditions. In addition to better understand stream exposures, we need to look at more than just water. We need to do multi-matrix sampling that includes water, bed sediment, and tissue. And because, as I mentioned, there's over 4,700 individual PFAS, we're never going to be able to measure everything. So let's use total organic fluorine as kind of a surrogate to see what, to better capture unidentified organic fluorine. And we're just developing, there's methods out there. We're developing our own organic fluorine method now, and that's what we're going to start using now as another tool in our toolbox to still look at our targeted suite of PFAS compounds, and hopefully that'll improve in sensitivity with time goes on and capture a broader net of PFAS, but complement that with total organic fluorine method to get that total fluorine exposures. And in the multi-matrix aspect that I mentioned previously, we already have that in the bag. We actually did a study last summer. We looked at 15 weightable streams across the state. Some of which were also already included in that recon that I talked about today. These were those that had a range of crop and livestock production. Again, sampled Muddy Creek and another stream that the basically wastewater treatment plant impacted sites. We went to a couple basins where we know there were substantial biosolids applied. And it's really hard to get a true reference site in Iowa, but we did go to a site in Northeast Iowa where there's lots of trees and just kind of have a minimally impacted site. That sampling, I said, as I mentioned, it's already in the bag. We collected all the samples last June. And this is a broad research team listed here. And again, it's a multi-matrix. So we did water, bed sediment, and minerals this time around. It's multi-contaminant. You see the range of contaminants we're looking at, including total fluorine that I mentioned. And it's a multi-lab effort from USGS labs to the University of Iowa, Greg Lefever's lab, the University of Iowa, the University of Illinois, measuring for microplastics. So again, taking advantage of really trying to see what is out there, not only just with PFAS, but these other contaminants, and see, is there a correlation? Is, for example, the more pharmaceuticals we see, does it have a positive or negative relation with PFAS and so forth? So there's a lot of little things we can do to hopefully better understand how PFAS and what PFAS are in our water resources. These are the references that I cited today. If you want any or all these papers, just let me know. I have paper copy or electronic copies I can share. So with that, I'll turn over to Matt with questions, and I'll throw up this information slide as I was asked. So Matt, I guess if we have time for questions, let's go into the question section.
We definitely have time for questions, and but first, if you are wanting a certified crop advisor continuing education credit, please email Liz Ripley at ejukums@iastate.edu by today at 5 o'clock with your name, the name you entered to watch the webinar, and your CCA number. So please do that. And maybe, Dana, you can click one more and we'll just put in a little plug for the next webinar. Okay. All right, next week we have Mark Lick, Dr. Mark Lick, that's extension cropping system specialist in our Department of Agronomy. Will be here talking about redefining the field edge case study. So this is kind of a last in a series on those case studies. Okay, so we do have a couple questions here. So the first one, Dana, is do you know of any efforts being developed to substitute for PFAS, more environmentally friendly or biodegradable rather than the PFAS
there
toxins?
Is, and hopefully it's going to go farther. And again, this is I'm not an expert, but what I've initially observed, it seems like part of what they're doing is just replacing one PFAS with another PFAS. I mean, they have all these beneficial properties that make them attractive for the various products we're talking about. But that's kind of the initial problem is, and I've seen some early results in studies that show these replacement PFAS can be just as problematic. I mean, those strong carbon fluorine bonds can cause just as much problem as what they replaced it with. So I don't want to call it a marketing ploy, but it is kind of a short-term solution that isn't really solving anything. I'm hoping, I can't give any specific examples, but I'm hoping people are trying to look at greener alternatives than just swapping out one PFAS for another.
Okay. Are there, great, thanks for that detailed answer. So next one, what are the most promising methods for treating or removal of PFAS from water? And when they volatilize, are they rendered less harmful or concentrated?
I'm not totally up on the treatment aspect. I do think there is some treatment that can remove these, whether that's activated carbon or reverse osmosis. So I think there is options out there, but all that comes at a price. I mean, if you have to upgrade your plant, either that comes at a price just with the physical upgrade. Some of these are pretty energy hungry technologies. So, we really, I mean, I can't say we need to go out there and upgrade every plant in the country, but there's options. But as always, treatment should be our last line of defense. We should never rely on treatment to be our solution. The real solution is, as we talked about in the previous question, let's find greener alternatives. Let's stop using these compounds if we can, at least to try to reduce the use, so that reduces the environmental input, because treatment is our last line of defense. We don't want to just keep going on our merry way and say, let's put all the effort into the treatment plant to solve this environmental problem. Now, the other question I think was with volatilization. I don't believe that volatilization render these compounds less of a problem. It just means they're going up into the atmosphere, traveling long distances, and then coming down either in the wet or the dry deposition. So it just means that these compounds, it's just another pathway for these compounds to be spread globally, essentially.
Next one, you may not know the answer to this, but very high levels of PFAS are being detected in Maine farmland. Is that because, or could that be because paper mill waste products were land applied?
That could be, and I'm trying to remember if this was with Maine or with Michigan. I remember there was one, and this I saw that either is, I don't think it's a journal article, just a science news story. And I saw it was related to biosolids applications. As I mentioned previously, like half the country's biosolids are land applied. There is beneficial source of nutrients in those biosolids, but that example I'm thinking of, they seem to point that back to biosolid applications, and then having problems with land applied to pastures associated with dairy, and then an uptake into the cows and then into the milk. I can't remember if that was with the paper mills that was mentioned, but it could be. But I certainly know the biosolid aspect has been a known problem in some parts of the country.
One is we're currently employing things like bioreactors or wetlands to treat drainage water. Do you think these would help or I guess enhance bioaccumulation of the PFAS?
Yeah, I don't know specifically. I mean, it'd be worth investigating. It'd be good to see, are those bioreactors actually causing chemical transformations and degradations? Now, they may be just slightly tweaked and still, that carbon fluorine binds, may still be impacted. So they may be a degradation product and still considered a PFAS. Or are these processes like a wetland? Is it just removing it from the water by simple partitioning to the sediment? So basically your wetland becomes an environmental sink. And that, basically the concentrations will build up in that wetland. And obviously wetlands are used for a whole host of organisms. So you may be solving or kind of helping one issue with the water, but causing another problem. So, there needs to be more investigated in terms of how beneficial that aspect would be.
I think one of your points was that there needs to be some additional research work on non-point source contributions here, and so better understanding what some of the transport pathways in the ag landscape seems like a potential researchable area.
Yep, exactly. And one, I mentioned a study from France that measured PFAS in livestock manure, and a study I didn't mention that we just wrapped up the sampling last month. We sampled, not a large number, but we sampled about 10 biosolids from across the country. We sampled about 10 to 12 livestock manures, and that included poultry manure, swine, dairy cattle, both organic and kind of a conventional dairy. We sampled a livestock yard, so we had cattle waste. So we'll get a better sense, and we sampled that for a whole range of contaminants, pharmaceuticals, antibiotics, pesticides, but we also included PFAS. So we'll get a better idea of what PFAS, what concentration levels are we finding in livestock manure. And, if we just find very low traces, that's one thing. If we find higher concentrations, then that leads us to kind of, look at what's going on in Iowa, because obviously livestock manure is being applied as a logical source of nutrients. But if there's PFAS in there, we need to understand what other consequences there may be from that land application.
Any idea on what the timeline to create regulations on PFAS in streams in the environment might be?
Ooh, I can't touch that one.
Okay.
USGS is a purely scientific organization. We have nothing to do. Fortunately for me, I don't want to get into the regulation policy aspect. That's a whole can of worms. That's really a tough nut. But hopefully the data we are generating feeds into the EPA and others that do make regulations. So that's the important aspect. The last thing we want to do is have an agency generate regulations based on emotions, because people hear these are forever compounds and say, we got to get them out, with really not understanding how prevalent they are or what concentration levels are of a consequence. So we need the scientific data to feed into them, but actual timelines and when this may happen. I do think, though, again, this is I'll put on my cap, and this is not a USGS official statement, but these compounds are a part of the USCMR 3s. I think there are more of them in the USCMR 4, and I think they're developing the USCMR 5 now. So, I mean, to me, I don't think there's been any new drinking water regulations since the 80s. But if something new is going to happen in terms of contaminants, I'm not a betting man, but I'd probably put a bet that anything new coming in terms of regulatory compounds are going to be a PFAS.
Next one, if there was a vegetable production on a local site that was a site of substantial land spreading and municipal sewage sludge, do you see a substantial risk to the consumer?
I don't know if I consider a risk. I think it's worth investigating. I don't know enough. I know other compounds, once you land apply, then the plants that you're growing in there could be uptake into the plants. I just don't know. I'm pretty sure that PFAS is likely going to be taken up, but is it going to be concentrated in the roots? Is it going to concentrate in the leaves? So will it be a part of the plant that humans consume, and it would be at high enough level? I wouldn't want to, sound sensationalized and say, wow, that's disastrous. But if we know that the source that's being land applied to whatever crop it is contains PFAS, it's worth investigating to, collect water sample, or collect the source being applied, collect soil samples before and after application, collect the plant and analyze various parts of the plants to understand, the distributions in the plant to see, is that something we need to be concerned about or not?
So there was a second part to that question, that if the situation described was an actual one, would this site be of research interest to you?
Oh, certainly. Yeah.
Okay.
Yeah, we have one example. Again, I couldn't get into everything we talked about today, but we have a research site, and actually it's not plot scale, they're field scale. They're about 15 to 18 acres at the university, or Oklahoma State University. So we had 3 fields where all have basically the same soils, all having the same agricultural practices. They planting with the same variety of corn at the same day, same herbicides are applied. The only differences of those 3 fields, one is basically a dryland corn. One is irrigated with municipal effluent, and it's only irrigated when moistures are, the crop is needing it. They're not just irrigating to irrigate, they're irrigating when the crops are needing more moisture. And one field is a biosolid amended field. And so we're collecting the source that's being applied, like I talked about before, whether it's effluent or biosolids. We're collecting the earthworms, we're collecting soil samples before and after. All those fields are instrumented. They're instrumented with weirs. So during rain-induced runoff, we're collecting the water samples over the storm hydrograph. So we're kind of doing that now. again, looking for all these different types of contaminants, including PFAS. But there's other examples where this is taking place. We're always, I can't say for sure we're going to zip out there and conduct research, but we couldn't do anything if we don't know about them. So certainly let me know, and we can see if this is something we can maybe fit within our research priorities.
I think you mentioned maybe some treatment technologies. Is there any biological way of treating for PFAS in any wastewater that you know of?
I just know enough about the details on the treatment side of things. I've kind of read a little bit here and there, but I'm just not sure which ones get. So I don't know if it's a activated sludge, if that makes a difference, and if in the longer contact time would improve removal or not. I don't want to misspeak on anything, so I'll leave it at that.
That's great. And do you know of work that's going on looking at kind of the impact of PFAS exposure on wildlife?
I've seen papers. I mean, again, I don't know the range of wildlife, but there's definitely papers that show that there is impact, whether that's aquatic bugs that are living in it. Is it something in fact of even higher organisms, or they're obviously we have corn-fed deer all over the state. Would that be an issue? So I can't give you a specific example of an organism that is having definitive impacts, but I've seen papers that show that we have enough information to know that there can be effects. But we don't know we know that, and that's the whole thing with water, with environmental quality in general. We have a hard enough time understanding the environmental consequences from a single contaminant. But in the real world, we know there's mixtures, not only mixtures of PFAS. We've seen as many as 9 PFAS in just this one study we looked at. And I know there's probably 10 to 15 other studies I've seen have 10 to 15 different PFAS or more. But it's the PFAS in the presence of pesticides or other organics. It's the challenge of, PFAS are surfactants. Does the presence of a surfactant enhance or change uptake to a contaminant more than it would have if those compounds were present? So that's kind of the $1 million question. I think that's the harder question. The easy question is, are these compounds out there? And that's difficult enough as it is. The real challenge is what's the environmental consequences from not only PFAS, but to mixtures of PFAS and everything else that's out there.
Right. Well, thanks so much, Dana. I think that addresses the questions that are kind of right up your alley. And so really appreciate your talk today. I think it's really important. And hopefully as more of your research becomes available, we can get you to come back and give another webinar. Just leave it to you. Any last comments?
No, I appreciate the opportunity to present just the paper that was published last fall. I'd be more than happy to keep you guys in the loop as I have more other papers published. I'm happy to repeat this again. And again, if you have any follow-up questions, feel free to email me. If you'd like to see any of the papers that I cited, just let me know and I'll fire it off on an electronic copy. So appreciate the opportunity to talk with everybody today.
Again, thanks so much, Dana. Liz did put the information in the chat box if you are looking for a CCA credit. And then just a little plug-in, come back next Wednesday at noon for Mark Licht talking about redefining the field edge case study. With that, everybody have a great rest of your day. Recording is stopped. Thanks so much, Dana. Good to see you.
All right. Thank you. All right.
Take care.
Bye.